To the in-person evolution seminar after this sequence of a month of virtual only talks. Nice to be here again. It's a pleasure to introduce today's speaker. Jered is a PhD student at the genetics program here and was a former ESS organizer. So when I joined the group, he was the one leading it. It's very nice to see what he's up to now.
All right, I'll minimize this quick. All right, thanks, Tiago. It's great to be back here and present to you all. So I'm a member of the PACER lab, and as such, I'm interested in the genetic processes that underlie evolutionary patterns. And specifically, I am interested in the genetic changes that occur during the adaptive process. And one of the most well-known examples of this process are organisms on islands. So islands are have always been a foundation for evolutionary study. They were the island forms that we see and their unusual phenotypes were key inspiration to Darwin and his theories of natural selection and have fascinated evolutionary biologists ever since then. So on these islands, we see forms that are unique in morphology and body size and life history and behavior. And the system I'm going to talk to you about today is no less exceptional in this regard, and that is the Gough Island house mouse. So Gough Island is a remote volcanic island isolated in the South Atlantic. And despite its extreme degree of isolation, a colony of house mice colonized this island roughly 500 years ago, probably as Europeans began exploring. So anyone who's familiar with these mice from my lab's previous work probably knows about them for their gigantism, a Gough Island mouse being roughly twice the size of a mainland house mouse. But this is not the only unique thing about these house mice. And it's actually not quite correct when I said in my title of my talk that this is a predator-free island. Because while there's nothing on this island that eats a house mouse, the mice themselves are predating on seabirds that use this island as a nesting ground. And this predation has gotten so prevalent that an eradication effort is actually undergoing right now to exterminate these mice from the island and protect the seabirds. But fear not, we have a colony of these mice here in Madison, and that provides a great tool for us to use as evolutionary biologists to study the genetics underlying all these unique phenotypes that are shown in these house mice. So these mice belong to the same subspecies as the laboratory house mouse, and as such, we have access to a whole wealth of genetic and biomedical tools that are available for us to study them. And in addition, aside from their predatory behavior that we see in these mice, there's plenty of conditions on the island that suggest to us behavioral evolution has occurred. So why in general do we expect behavior to evolve on islands? There are several reasons. I'm listing just a couple here. So first of all, organisms on islands often have a lack of predators, which drastically changes the risk foraging assessment that these organisms need to decide on when trying to balance their danger to themselves and the necessity to acquire food and shelter, so on and so forth. In addition, islands often have a very different set of natural resources that these organisms need to go and acquire. And for house mouse, as the name might imply, they typically live in houses, or at least barns, or something man-made, none of which occur on Gough Island. So for a Gough Island house mouse, they have very different sets of resources, both food and shelter, that they need to go out and find than what is common for a mainland house mouse that has evolved alongside humans for a very long period of time. So for these reasons, we predict several different things to have evolved in this setting, and that includes increased boldness, increased exploration, and reduced predator avoidance when a predator cue is re-exposed to this island population of house mice. So collectively, these behaviors have sometimes been called as island tameness. This is something Darwin has referred to, and is something that many other evolutionary biologists have experienced before, including Peter Grant, as we see in this example here. So all of this leads to the main question that I'm going to address in my talk today. Are there heritable differences in behavior between Gough Island and mainland mice? So this is how I set out to address this question. So at our colony here in Madison, we have inbred strains of Gough Island mice and mainland mice, all of which live in constant environmental conditions. So this means the differences we see between these two strains come primarily from a genetic component. So in this situation, all of these mice from both strains are born into their parents' cages. They're weaned at 3 weeks old into cages with one litter mate of the same sex. 1 week after this weaning, they get their first behavioral test, which is this open field. And this is a behavioral assay for anxiety at the prematurity stage. So 4 weeks after that, after puberty has occurred, they get re-exposed to this open field test, followed by a light dark box test and a predator cue test at 9 and 10 weeks old, respectively. And I'll go through the details of each of these tests in a moment. So all of this together provides a deep behavioral profile for both strains of these mice at multiple life stages and across multiple simulated environmental conditions. So here are just the arenas I use in these separate tests. So on the top left here, we see the open field box. This is just a very simple, bright open box. On the right, we see a light dark box. This is similar to the open field, except now half of it is shaded, which provides the simulated shelter for a mouse to go into if they choose. And in the bottom, we have a Y maze that I use in a predator cue test, each arm of which you can place distinct cues into and see where the mice prefer to go. So I'll start by looking at this. I'll start by summarizing how each of these tests is conducted. So you can't quite see her at the top, but the first thing that you do is you take each cage of these two mice each, put them into the behavioral testing room, which is separate from the main colony. The mice in that cage are allowed to acclimate to that room for 30 minutes. You then take the first subject from the cage, place it in the respective test chamber for that behavioral test, then video record their movement for 30 minutes, completely undisturbed. You then take the mouse back, return it to its cage, and count the number of fecal boli it left behind, which is an indirect measure of anxiety. You then repeat these 3 steps here for the cage mate. So this is a survey of what happens after this test. So after every test, you now have a video of what the mouse was doing during that time. And in the modern age of computers, we can acquire consistent and unbiased behavioral estimations through automated procedures. So here's an example of open field test that I conducted. I've included the audio in here so you can get an idea of what's going on. That sound that you hear is actually an air vent in that room, which provides constant white noise, which is something that the behavioral core on campus suggested was actually a very good thing for these kind of tests because it blankets out lots of background noise. So this is a consistent noise that you hear, and I'll block it out now for you as I explain the rest. So I'll let this video keep running. You can see the idea of what this mouse is doing. So this video is then transferred through an ImageJ macro script, which was associated with a different software called Mouse Move, which I heavily modified, all of which just condenses this video into a series of observations of the X and Y coordinates of where that mouse was for every frame of the video. This data is then fed through a series of Python scripts that I developed, and this pulls out the actual behavioral measurements that we're interested in. So before I get into the actual measurements, I wanted to provide a couple of broad surveys or broad summaries of what these behaviors are that hopefully will stick into your mind. So this is the image I included on the poster for this talk, if you happen to see it. And many of you are probably wondering what the heck was going on. So this is actually a favorite in the lab. And if there's one thing you can take away from this talk, I hope this sticks in your mind. So this is just summaries of where these mice, 2 individuals, went for a half hour in the open field test completely undisturbed. So this one on the right is a mainland house mouse. And you can see it primarily hugs around the edge of the open field with some crossings through the center. On the left is a Gough Island mouse. And you see it really did its best to cover as much of the box as it possibly could during this half-hour test. So these were just two individuals. This is actually the first ones that I developed these plots for from the two respective strains. But you can also summarize this across all of the mice that I tested. So that's what this heat map is. So this is now summarizing all of the mice for the adult stage during this open field test in these two respective strains. Color doesn't show up quite as well on the auditorium screen here, but hopefully you'll still see that this mainland strain is much darker throughout the center with some peaks along each of the corners and lines connecting them, while the Gough Island map is much more uniform throughout. So this is exactly what you would predict for a mainland mouse that is highly anxious. It tends to prefer the corners, places where it's much less likely to be detected, compared to a Gough Island mouse, which is much more bold and much more exploratory and more active. Okay, so let's get into the actual data and what I measured. So for the open field test, there are plenty of predictions that have been developed for us in studies using the laboratory mouse for people who are developing anxiety related drugs or therapeutics, that sort of thing. So we can draw on all of their findings to make some predictions here. So for a bold, active mouse, we expect it to spend more time in the center of an open field, which I computationally define here. It's this circle with diameter half the length of a side of the open field. We also expect these mice to travel much more overall and to leave a fewer number of fecal boli after the test. So let's start with that data for the fecal boli. This is exactly what it looks like. So after the mouse is done with this test, it left various numbers of fecal boli behind. You go by and you just count all of those. And what we see is that Gough mice are meeting our expectation that they are less anxious than our mainland mice. So at both life stages here, Gough mice are leaving fewer number of fecal boli behind than the mainland mice. I'm including the sexes here just for your reference, but throughout all of these traits, very few sex effects were detected, and if they were pretty minor. Okay, so for time and center, we see a very similar picture. These Gough Island mice at both life stages spending much more time in the center of this open field than our mainland strain. When you're looking at the distance traveled, you get a slightly different picture. Gough mice are traveling more overall than mainland mice, but this only occurs at the adult life stage. So now you're seeing a life stage component affecting this trait, which suggests distance traveled, which I am compounding with exploratory behavior, is uncoupled from these anxiety-related traits that I just showed you previously. So I also had the chance to measure some F1s that come from crosses between Gough and mainland mice. So this is the exact same data I just showed you, including observations from these F1 individuals. So you see the juveniles over on the left, adults on the right. Data for time and center and distance traveled for the Gough and mainland strains that I just showed you, but now including the F1s come from either maternal cross direction, either a Gough mother or a mainland mother, including here in the middle. So we can find a couple of interesting things from these F1 distributions. First of which, that there is a subtle but significant cross direction effect detected for distance traveled. So F1s that were raised by a Gough mother travel slightly less than F1s raised by a mainland mother, despite their genetics being pretty much the same, except for differences on sex chromosomes, mitochondria, that sort of thing. So again, we see this observation that distance traveled, this exploratory behavior, is getting separate inputs that these anxiety-related traits are not getting. Additionally, throughout all of our traits, we see pervasive dominance of F1s clumping towards the mainland strain. So this is just summarizing what I just told you. So for the open field test of both juvenile and adult stages, we see that distance traveled, this exploratory behavior, has effects from both the cross direction and age, a slight sex effect, while these anxiety-related traits don't receive any of that. They are consistent throughout all of these different factors. So it suggests exploration and anxiety-related traits are uncoupled in the system. Additionally, we see pervasive dominance, as I just told you, mostly occurring towards the mainland strain. For most of these traits, the F1s group statistically at that point halfway between the mid-parent and the mainland, which I call this mid-parent mainland midpoint. There's one notable exception, which is the number of fecal boli. At the juvenile stage, F1s actually clump with the Goff strain, whereas adult F1s clump at the mid-parent value. So even though the number of fecal boli matches the same expectations we have for anxiety being reduced in Goff mice and increased in mainland mice, the genetics underlying this trait is slightly different and may have to be involved with things related to body size that is increased in Goff mice, morphology of the gut, so on and so forth. Now, despite that difference, the dominant pattern we see overall across these traits suggests that these behaviors might have a simpler genetic basis than some other kinds of complex traits, like body size. And that suggests an increased chance of finding the causative loci underneath these adaptations. And I'll get into that later during my talk. Sorry, you can't see the title there, but the other thing we wanted to measure in order to get a separate measurement or assessment of anxiety and how it's context dependent is to see what happens when these mice have access to a potential shelter. So to do that, we use a light dark box. As I showed you before, this is just a simple box, except half of it is now shaded. Mice still get free run of the entire arena and can choose whether they spend their time in the sheltered component as opposed to this exposed component. So you would expect for a highly anxious mouse, such as the mainland mouse, it would prefer the sheltered component because it has a less chance of being detected by a potential predator. And this is exactly what we see. So mainland mice overall during this test are spending more time in the sheltered component, while Goff mice are more willing to travel into this light chamber. You also see that just the number of entrances into this light chamber is increased in Goff mice, which harkens back to this increased distance traveled we saw in the open field. There's a lot more crossings, a lot more movement overall in Goff mice. So this general pattern still holds when you draw a new threshold at the halfway point for this light chamber. So Goff mice are still willing to go farther towards the edge of this light chamber at an increased distance from the shelter. So they're using the whole reign of the arena. Now, interestingly, this being a half-hour test, if an object were to randomly move throughout this whole arena, you would expect it to spend half of its time in the light chamber, half in the dark, which is this line I'm putting right here. So Goff mice are getting pretty close to this level that we're seeing. So there is still evidence that they prefer the dark chamber, but they're pretty close to neutrality in having no preference between a light or a dark sheltered arena. Okay, so the last test I did was to see if Goff mice still respond to a direct predator cue. So to do this, I used the Y maze. Mice are first put in this bottom arm here, which I'll refer to as the blank arm. They get 10 minutes to fully explore this arena without any cues present in order to get a baseline for arm preference. After that point, the mice are shooed back into this corner. This barrier is inserted. And then I put a predator cue in one arm, which is a cotton ball soaked in fox urine. Red foxes being a very common predator of house mice across the entire world. And the other arm, you get this control cue, which is a cotton ball soaked in deer urine, deer having pretty much no interaction with house mice. And so we expect for a cue that mice don't really care about, you would spend equal time in that arm with that control cue compared to the blank arm. this ratio ends up as 1, and we would say there's no avoidance to that Q. Alternatively, if mice are spending less time around the predator Q compared to the blank arm, this ratio becomes less than 1, and we say that they are avoiding that predator-associated Q. So this is what that data looks like. So in blue here, you're seeing this ratio being plotted out for both strains with respect to the deer urine control Q. And in red, you see that ratio plotted out for the fox urine Q. Triangles are Goff, squares are mainland mice. But the main pattern that you'll see here is there's pretty much no difference between our two strains in this trait. So before any Qs are placed, pretty much all of these ratios are the same. There's no statistical difference between any of these. But once the Qs are added, you see avoidance with respect to the predator Q in both Goff and mainland mice. So in this specific case, when there is evidence of a direct predator Q, Goff mice still retain this avoidance response that you see in mainland mice. Additionally, if you measure the level of stress hormones in these mice immediately following this test, all of these mice are pretty stressed overall. So this assay actually caps out at 1,000. So all of these levels are high to extremely high, meaning these mice were highly stressed after being exposed to this predator Q. Okay, so to summarize, we had several predictions for a bold and active mouse that we get from laboratory strains for all these tests. So we expect for our Goff mice, if they evolved along these lines, to spend more time in the center of an open field, to travel more overall, to leave fewer numbers of fecal boli behind, to spend less time in a shelter if one was available, and to spend more time around a predator Q if one was placed there. And we saw this was consistent for almost all of these traits, but they have retained this avoidance response for a very direct predator Q. So we see, again, the context-dependent nature of this behavioral evolution. So mice, in a situation where there's potentially predators around, but you have no direct evidence for it, Goff mice really don't care. They'll travel wherever they want, where mainland mice still want to reduce their chances of detection. However, when you place a direct predator Q in there, Goff mice still are very much afraid. So all this together suggests Goff mice have heritable changes in behavior that are associated with island life, but they have retained an avoidance to direct predator Qs. We also saw that exploratory related traits are influenced by age, sex, and cross-direction, while boldness is not. And we saw from F1 distributions that there's pervasive dominance throughout all of these traits. And that suggests increased success for identifying causative loci underlying these different adaptations. So that is the next main goal for my research. So to address that, I undertook a genetic cross. This is what that design looks like. So you cross Goff and mainland mice together to generate these F1s in both maternal directions, just as I did before when I showed those F1 distributions. You can then take those F1s and mate them together in order to get F2s. And this shuffles up the whole genome, which allows you to look for associations between specific regions of the genome and these different behaviors. So I collected data from over 600 genetically unique F2 individuals. That includes behavioral phenotyping in the same manner as the mice that I just showed you, going across both open field tests and the light dark test. I combine this with genotype data that comes from a microarray that's designed to capture diversity among house mice. So it provides a very rich set of genetic data between our Goff and mainland strains. So by combining these two, we can look for those associations between regions of the genome and these different behaviors in the same manner that previous researchers have done for pretty much the same exact traits in the laboratory mouse. So that's what this plot here on the left is looking at. So this association, which is shown here as something called a LOD score, you have associations between a region of the genome and these different kinds of behaviors, such as open field activity, defecation scores, so on and so forth. In addition, Mark Nolte, a different member of the lab, also collected a very ambitious data set that provides lots of context to help us find specific genes associated with these behaviors. So for a subset of these 600 F2s, following the final behavioral tests, he rapidly dissected out the brain and isolated specific regions that are known to be involved in fear and anxiety. From that, we're hoping to collect RNA-seq data from those regions, and that'll help us find associations between the expression level of certain genes and these different kinds of behaviors. A hypothetical example being put here, where as the expression level of a certain gene increases, it spends more time, say, in the light side of the light dark box. So all of this together provides us many different avenues in order to narrow down on the exact regions of the genome that are underlying these different behavioral traits. So a lot of this data collection has already occurred, and now we're in the analysis stage, so please stay tuned. Hopefully we'll have some new findings for you in the near future. So finally, I just want to mention one additional thing that I'm hoping to do, and that's to look for indirect genetic effects underlying these different behavioral traits as well. So an indirect genetic effect can be explained as an environmental influence on the phenotype of one individual that is due to the expression of genes in a different conspecific individual. So mathematically, it looks something like this. So in the standard case, you have some phenotype that you're interested in, and that's made up of a genetic and environmental component. But in this context, this individual, the phenotype that we're measuring, can also be influenced by an interaction with a conspecific individual, that individual's behavior also being made up of a genetic and environmental component. So this sub-environmental component that this individual, focal individual, is experiencing also has a genetic component to it and is thus evolvable. So that is something we can look for in our context as well. So here's a little more concrete example. This was a study conducted in deer mice looking at aggressive behavior. So in the traditional case, you have this focal individual. This is the one you're actually measuring. It has an environmental and a genetic component to that. And then you have whatever opponent it's interacting with. In an indirect genetic effect context, you're also considering what is the genetic component of this opponent, genetic component of this opponent that's interacting with this focal individual, and is there some kind of detectable genetic effect that's influencing the behavior of who you're measuring. So in our context, all of our mice were raised in cages with one litter mate of the same sex, and in F2s, each of these is genetically unique. So in the traditional QTL mapping sense, you have the phenotype of cage mate A, you take the genotype of cage mate A and you look for associations. And you do the same thing using the phenotype of B, the genotype of B. In our situation, since there's consistent caging arrangements throughout this whole study, we can flip this on its head and say, is the phenotype of mouse A influenced by the genotype of mouse B, its cage mate, and vice versa? So looking for this, now we're saying, are there associations in the genome that affect the behavior of the mouse you're interacting with? So with that said, I have lots of people I want to acknowledge. Brett has been a wonderful advisor and very encouraging throughout this whole project. Mark, as I mentioned, has been very involved and is doing his own avenue with this research. An undergraduate did the very first pilot study showing behavioral differences existed between these mice. So Megan, I want to acknowledge as well. I'd like to thank everyone who's worked in our colony, my whole advisory committee, my funding sources, and my older brother, who was very helpful in developing these computational pipelines in order to analyze these videos and collect behavioral data. So with that said, I think I have plenty of time left for questions, and thank you for listening.